Hello everyone, my name is Hussein and our today topic is about hyperkalemia. Hyper means high, mia means blood, hyperkalemia means high concentration of potassium within the blood, and I try to present this topic on three separate video, and the first video will focus on the physiology of potassium, and the second video will focus on the cause and clinical manifestation of hyperkalemia, and the third video will focus on the treatment of hyperkalemia. When we see the physiology of potassium, our body stores around 3000 milliequivalents or 50 to 75 milliequivalent of potassium per kg. And from these 3000 milliequivalent, around 98% of potassium stored within the intracellular fluid and make the intracellular fluid potassium concentration around 140 milliequivalents per liter, and the remaining 2% stored within the extracellular fluid and make the extracellular fluid potassium concentration around 4 to 5 milliequivalent per liter. When we say hyperkalemia is when the extracellular fluid potassium concentration greater than 5 milliequivalent per liter. But hyperkalemia can be divided into mild, moderate, and severe based on the degree of potassium. So when the potassium concentration is between 5 to 5.5 milliequivalent per liter, it is called mild, and between 5.5 to 6.5 milliequivalent per liter, it is moderate, and between when the potassium concentration greater than 6.5 milliequivalent per liter, it is severe. But hyperkalemia is depending on the concentration of potassium in the extracellular fluid, not in the intracellular fluid. So anything that increases the movement of potassium from the intracellular fluid to the extracellular fluid can lead to hyperkalemia. The other very important concept is the resting membrane potential. The ratio of potassium in the intracellular fluid and extracellular fluid is very important to determine resting membrane potential. So the effect of if you understand the effect of hyperkalemia in the determination of resting membrane potential, you will easily understand the clinical manifestation of hyperkalemia on cardiac and skeletal muscles. So, for example, when we see the resting membrane potential of large nerve fibers, it is around minus 90 millivolts. That means the inside of the fiber is 90 millivolts more negative than the outside of the nerve. What makes the intracellular environment more negative than the outside environment? There is two responsible body. The one is the sodium potassium lead channel, and the second one is the sodium potassium pump. And how they maintain the intracellular fluid compartment more negative than the extracellular compartment. We'll discuss on the next slide. For example, if it is a cell and within the cell the predominant cation is potassium and the predominant negative ion is protein and phosphate. And in the extracellular fluid, the predominant positive ion is sodium and the negative ion is chlorine followed by bicarbonate. So if it is a sodium lead channel, it moves sodium and potassium from the region of high concentration to the region of low concentration. So this sodium potassium lead channel moves potassium from the inside to the outside, and it also moves sodium from the outside to the inside. But the permeability for potassium is 100 times the permeability for sodium. So it moves more potassium from the inside to the outside, and also it moves a small amount of sodium from the outside to the inside. So this sodium potassium lead channel causes the movement of excessive potassium from the inside to the outside, and it can create a relatively negative environment in the intracellular compartment because of loss of more positive charged potassium ions. So this sodium potassium lead channel creates a resting membrane potential of minus 86 millivolts. The other very important pump is the sodium potassium pump. This pump uses ATP, and when ATP breaks down into ADP and inorganic phosphate, the channel become open and move sodium from the inside to the outside, and also it moves to potassium from the outside to the inside. So three positive charges moves from the inside to the outside, and two positive charges move from the outside to the inside. So it can cause the movement of any one positive charges from the inside to the outside, and it creates a relatively negative environment, and it creates about minus 4 millivolts. So the minus 4 millivolts that is created by sodium potassium pump, and the negative 86 millivolts that is created by potassium sodium lead channel creates a resting membrane potential of minus 90 millivolt. So this 90 millivolt is the resting membrane potential of large nerve fibers. And the other very important concept is how hyperkalemia affects this resting membrane potential. For example, in case of hyperkalemia, there is high concentration of potassium in the extracellular fluid. So the concentration of potassium in the extracellular fluid and the concentration of potassium in the intracellular fluid become more balanced. So there is no movement of potassium from the intracellular compartment to the outer part of the cell. So the movement of potassium from the inside to the outside, so the lead channel will decrease, and that can lead to the accumulation of positive potassium channels inside the cell. So when these potassium channels accumulate within the cell, it decrease the resting membrane potential from 90 millivolts to minus 70 millivolts. So when the resting membrane potential decreases from 90 millivolt to minus 70 millivolt, it can cause the opening of the voltage gathered sodium channels with due to small stimuli. So when the voltage gathered sodium channels become open, there is influx of sodium from the outside to the inside, and that can create an action potential. So action potential will be occurred with a small stimuli, and that can lead to activation of the cells. So when there is hyperkalemia makes the resting membrane potential less electronegative. So that means it changes from minus 90 millivolt to minus 70 millivolt, and that can lead to partial depolarization of the cell membrane, and a less depolarizing stimulus is required to generate action potential, and that can increase the membrane excitability. So, but the persistent depolarization can inactive the sodium channels. So when the sodium channels become inactive, there is a net decrease in the membrane excitability, and this decreased membrane excitability can lead to impaired cardiac conduction and neuromuscular weakness. So in the early phase of hyperkalemia, the resting membrane potential decrease from 90 millivolts to minus 70 millivolts, and that can lead to the opening of sodium channels within small stimuli, and that can generate action potential, and that can activate the membrane. But after some period of time, or after persistent depolarization, the sodium channels become inactive, and that can lead to decrease in the membrane excitability, and that can affect the conduction of cardiac muscles, and also affect the neuromuscular junction, and the hyperkalemia will manifest with the sign and symptom of cardiac and skeletal muscle abnormality, and the patient will develop cardiac abnormality and also neuromuscular weakness and paralysis. So the other effect of hyperkalemia is on HERG gene. HERG gene is a gene that is encoded for HREG channel. This RHG channel is easily activated by hyperkalemia and inhibited by hypokalemia. And this HERG gene is responsible for the efflux of potassium during the time of repolarization. So during repolarization, there is efflux of potassium from the cell to the outside of the environment. So when there is hyperkalemia activates these channels, and when this channel activated, potassium moves from the inside to the outside environment during repolarization, and the repolarization becomes rapid. So when we see an ECG, the repolarization phase is represented by the T wave, and due to rapid repolarization of a cardiac muscle due to hyperkalemia, the T wave become peaked and tall T wave, and this T wave become ticked, and the and also it become narrow, and the distance from the Q and the distance between Q and T become short, and so the early sign of hyperkalemia on the ECG could be peak QF because of rapid repolarization, and the QT interval becomes short. So it is the early sign of hyperkalemia. And what are the other signs of hyperkalemia on the ECG? We will discuss in the next video, especially in the cardinal manifestation or in the clinical manifestation of hyperkalemia. So the other factor is a factor that affecting the concentration of potassium in the extracellular fluid. So the concentration of potassium in the extracellular fluid can be determined by certain things. The first one is potassium intake. Potassium intake is not a common cause of hyperkalemia, but it can affect the concentration of potassium in the extracellular fluid, but it's not common cause because when you eat excess amount of potassium, some of the potassium will store in our cells and some will excrete through the urine. So potassium intake is not a common cause of hyperkalemia. The other factor that affecting the concentration of potassium is extracellular fluid is the distribution of potassium between intracellular fluid and extracellular fluid, and any factor that increase the distribution or the movement of potassium from the cell to the extracellular fluid can lead to raise in the extracellular fluid potassium, and that can lead to transit elevation or transit hyperkalemia. Any factor that move potassium from the extracellular fluid to the intracellular fluid can treat a patient with hyperkalemia. So there are many factors that affect the distribution of potassium between intracellular fluid and extracellular fluid, and we'll discuss in the next video. The other factors that are affecting the concentration of potassium in extracellular fluid is the urinary potassium excretion, and anything that increases excretion of potassium through the urine can treat a patient with hyperkalemia, and any factor that decrease the excretion of potassium through the urine can lead to persistent hyperkalemia. For example, CKD and AKI decrease the excretion of potassium through the urine and can lead to persistent hyperkalemia. So, what are the factors that affect the distribution of potassium between intracellular fluid and extracellular fluid? One is insulin, and one is beta-2 receptor agonists like the salbutamol and the trauma and metabolic acidosis. They affect the distribution of potassium between intracellular fluid and extracellular fluid. So, for example, if it is a cell and in the cell there is one important pump, the sodium potassium pump. The sodium potassium pump uses ATP, and when ATP breaks down into ADP and inorganic phosphate, it moves 3 sodium from the inside to the outside and 2 potassium from the outside to the inside. So overactivity of this pump can lead to the movement of potassium from the outside to the inside, and it can create a patient with hyperkalemia. What factor activates this pump? One is insulin, the other is beta-2 receptor like salbutamol activates these pumps and moves potassium from the outside to the inside and create a patient with hyperkalemia. What factor inhibits this pump? The digoxin and the fluoride inhibit this pump, and if this pump is inhibited, no movement of potassium from the outside to the inside, so can lead to hyperkalemia in case of intoxication by digoxin and fluoride ions. And the other is the mechanism of action of digoxin. This digoxin is very important drug for the treatment of a patient with heart failure. This digoxin inhibits the sodium potassium pumps, and if this pump is inhibited, there is no movement of sodium from the inside to the outside, and also no movement of potassium from the outside to the inside. So the if no movement of sodium from the inside to the outside, the concentration of potassium is outside will decrease. So there is another exchanger, the sodium and calcium exchanger. When the movement of sodium from the outside to the inside, from high concentration to the low concentration, it creates energy, and that energy is very important for the movement of calcium from the inside to the outside. So if digoxin inhibits this sodium potassium pump, no movement of sodium from the inside to the outside, and the outside sodium decrease. If the outside sodium decrease, no movement of sodium from the outside to the inside, so there is no energy that brings calcium from the inside to the outside. So calcium will store within the cell, and the calcium that stored in the cardiac cell increase the contraction of the cardiac cells and can treat a patient with high with heart failure. So digoxin is one of the treatment for. So digoxin increase the concentration of calcium within the cardiac muscles and increase the force of contraction of the failed heart and improve the sign and symptom of heart failure. The other effect of digoxin is increase the activity of parasympathetic nervous system in the heart and decrease heart rate. So digoxin decrease heart rate but increase concentration of the calcium within the cell and that increase the force of contraction of the heart. So it improves the sign and symptoms of heart failure. So the other cause of hyperkalemia is metabolic acidosis. When there is metabolic acidosis, there is an increased proton or hydrogen ion in the extracellular fluid, but this hydrogen ion moves from the extracellular fluid to the intracellular fluid to bind with a negative charged proteins within the cell. But when there is a movement of hydrogen from the extracellular fluid to the intracellular fluid, there should be a movement of potassium from the intracellular fluid to extracellular fluid, and that can lead to hyperkalemia. So there are two types of metabolic acidosis. One is the anion gap metabolic acidosis, and the other is non-anion gap metabolic acidosis. The exchange of hydrogen or proton and uh, 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 the exchange between the proton and the potassium is only in the case of non-anion gap metabolic acidosis. But in the case of anion gap metabolic acidosis like diabetic ketoacidosis and lactic acidosis, the hydrogen that is moved from the extracellular fluid to the intracellular fluid will accompany with negative charged molecules, and so there is no exchange of hydrogen and potassium between the cell and the extracellular fluid. So non-anion gap metabolic acidosis is a cause of efflux of potassium from the inside to the outside. But in the case of diabetic ketoacidosis, we can get hyperkalemia, but hyperkalemia is not due to the exchange between hydrogen and potassium, but the cause of hyperkalemia in case of diabetic ketoacidosis is due to two factors. One is in the case of diabetic ketoacidosis, there is a deficiency of insulin. If there is a deficiency of insulin, the sodium potassium ATPase activity will decrease, and if the sodium potassium ATPase activity decrease, there is no movement of potassium from the outside to the inside, and that can lead to hyperkalemia. The other effect of or the other the cause of hyperkalemia in case of diabetic ketoacidosis is hyperglycemia. When there is hyperglycemia, when there is glucose, high level of glucose in the extracellular fluid, there is a movement of water from the cell to the glucose, or the water moves from the cell to the extracellular environment due to osmotic gradient. So when there is a movement of water from the inside to the outside, there is an increased concentration of potassium within the cell, and this high concentration of potassium within the cell creates a concentration gradient and can cause a movement of potassium from the inside to the outside. And the other cause of hyperkalemia is when there is a movement of water from the inside to the outside environment, the water carry potassium and the potassium will increase in the extracellular environment. So diabetic ketoacidosis increase or can lead to hyperkalemia by two mechanisms. One is due to insulin deficiency, the other is due to hyperglycemia, but not due to the exchange of hydrogen and potassium. The other cause of hyperkalemia, the other very important cause of hyperkalemia is trauma. Trauma, when there is an injury to the cell and cell membrane, there is an efflux of potassium from the inside to the outside. So any factor that can damage the cell membrane can lead to hyperkalemia. And the other very important cause of hyperkalemia is a factor that decreases the excretion of potassium through the urine. There is one important cell in the renal tubes that is called the principal cell, which is found in the connecting segment and cortical collecting tubules. And this principal cell absorbs sodium and secretes potassium. How? We'll discuss in the next slide. For example, if it is a renal tubulus, and within the renal tubulus the sodium and the water moves from the proximal tubulus to the distal tubulus. But if it is a principal cell that is found in the tubulus, it has apical side and basolateral side. In the apical side there is a sodium channel, and in the in the apical side there is sodium channel, and in the basolateral side there is sodium potassium ATPase, and also it has potassium channels. So when there is a movement of water and sodium through the tubules, the sodium moves from the tubules to the principal cells through this sodium channel, because within the cell sodium is in low concentration, so sodium moves from high concentration to the low concentration through this channel. But when there is a movement of sodium from the tubules to the principal cells, the delivery of sodium from the proximal tubule to the distal tubules will decrease. So the concentration of positive charge in the distal part of the tubule will decrease. So the distal part become more negative. And the sodium that is moved from the tubules to the principal cell also move towards the interstitial fluid or towards the capillary by sodium potassium ATPase. This sodium potassium ATPase use ATP, and at that time sodium moves from the principal cell to the interstitial fluid, and also the potassium moves from the interstitial fluid to the principal cells by sodium potassium ATPase pump. The movement of potassium from the interstitial fluid to the principal cell will increase the concentration of potassium within the principal cell. And when we discuss in the movement of sodium, the sodium moves from the proximal tubules to these sodium channels, and the distal tubules become more negative, and this negative environment attracts the positive charged potassium from the cell to the tubules. So the potassium moves through this channel into the tubules and excreted through the urine. So any factors that affecting this process can lead to hyperkalemia. For example, if there is a factor that affecting the delivery of water and sodium through the tubules can cause hyperkalemia. Example, if there is heart failure, GI dose or renal dose, there is a decrease in the delivery of water and sodium through the tubules, and that can lead to hyperkalemia. And there is also another important factor that's called the aldosterone, which is secreted from the adrenal gland. This aldosterone increases the number of the sodium channels and also the sodium potassium ATPase pump, and that can accelerate the excretion or the secretion of potassium, and also accelerate the reabsorption of sodium. So aldosterone increases the number of sodium channels and the sodium potassium ATPase pump. So aldosterone is very important factors that affecting the concentration of potassium. So any drugs that inhibit the sodium channel, and also any drugs that inhibits the aldosterone activity can lead to hyperkalemia. So amylorides and trimetrin inhibit the sodium channels, and also there is another drug that is spironolactone and epinol that is aldosterone antagonist, and the both of the drugs can lead to hyperkalemia. So potassium sparing diuretics divided into two, that is amyloride and trimetrin that directly inhibit the sodium channels, and the other is aldosterone antagonist like spironolactone and epinol. So it's all about physiology of potassium, and we will discuss the cause and clinical manifestation in treatment of hyperkalemia in the next video. And if you like this video, please subscribe, like, and share.